now we're going to look at non-coding RNA molecules. So remember that DNA can either, or a gene, can either code for a protein or code for an RNA. So we have RNA genes. Interestingly, only about 20% of the RNAs in the body actually code for proteins. The other 80% are these functional RNAs that we mentioned previously. Okay? So RNAs are very important, even though they tend to be an afterthought. These RNA molecules can be classified in several different ways. So I've tried to show you maybe 3 function organisms they exist in and the size. So this first thing is showing us just some. This isn't all inclusive of the ones we'll talk about. Some are in prokaryotes and eukaryotes. So RNA, tRNA, some cytoplasmic RNAs. Some, the majority of them, exist only in eukaryotes because they are more complex. Some are only in prokaryotes, with the example being CRISPR. And then we also have viral DNA. They have the ability to bind to different molecules. So the red are our non-coding RNAs. They can bind to a DNA molecule. Okay, so the first one you see is a non-coding RNA molecule that is complementary to a DNA strand. They can bind to mRNA, so we'll see now that they are non-coding RNAs that bind to mRNA and stop the mRNA from doing its job. They can bind to a protein or even small molecules. Then we have some that can interact with several different molecules. Okay, so multiple binding sites for proteins. This all affects their job, their ability to do their job. Now, in addition to how they bind, there are different sets of functions that they can have. So some are scaffolding. So back to the same picture where it binds multiple ones, it serves its purpose to form a complex. Others are guide. So it comes and takes a molecule from one place to another. And we'll look at several examples of this. And in order for this to occur, that RNA molecule has to have a binding site for a protein as well as for a DNA area. Some of them alter the function of a protein or affect its stability. So it can go in and alter the structure, which we know alters the function. So it can affect the ability of a protein to act as a catalyst. It can affect the ability of a protein to bind to another protein or to DNA, and it can affect the stability of that protein. Some of them function as ribozymes. So remember RNAs with catalytic functions, and we talked about this when we talked about tRNA and rRNA molecules. Some serve as a blocker, so they physically block a process from happening. And then some are decoys, so they recognize other non-coding RNAs and they stop them from working. Okay. The other categories, so I told you according to size. So some of them are considered long non-coding RNAs if they are greater than 200 nucleotides in length. Some of them are considered short regulatory RNAs because they are below 200. So in the next chart that we look at, you'll see that they are color-coded. If they are red, they represent the long non-codings. If they are blue, they represent the small regulatory. If they are purple, they can exist in both. So depending on what the molecule is, it can be greater than or less than 200 nucleotides. And then some I have classified as neither category. Okay, so here's the first set. So we have telomerase exist, hot air, RNase P, and small nuclear. This color needs to change. That represent the long, and then these represent the shorter ones. And we'll go through each of these. And there's 2 charts that we'll talk about in 2 separate videos. So telomerase, we talked about telomerase. So telomerase is an interesting molecule in that it has a protein and an RNA section. So the RNA section of telomerase, its job is to serve as a template for building telomeres at the end of proteins. We have exist, so we have talked about exist time and time again. exists as a long non-coding RNA that's complementary to the X chromosome, and its job is to coat that X, helping us to inactivate it. Hot air is new. So hot air was recently discovered. It's a hoax transcript antisense intergenic RNA, all right? It alters the chromatin structure, thereby repressing gene transcription. So here we have our hot air non-coding RNA. It binds to these complexes of protein. We see two of them here. Then it binds to a GA-rich region of its target gene. So here's the GA-rich region, and we see it nicely fits in there, and here's the target gene. Then it allows this complex of proteins on it to methylate, okay, and to demethylate areas. And we know that those histone modifications now are going to inhibit transcription. So hot air binds to a GA-rich region upstream of its target gene and causes methylation of some of the histones and demethylation of others, thereby repressing transcription. We have RNase P. We've seen RNase P when we talked about making a mature tRNA. So RNase P is an endonuclease that goes in and cuts the tRNA strand. Small nuclear RNAs serve as a part of the SNRP complex, so they are in splicing. So small nuclear RNAs, ncRNAs, their job is in splicing. And which RNA molecules are spliced? You should know the answer to that. Small nucleolar RNAs, their job is to help in cleaving the rRNA, so in processing it. So first you get the large rRNA molecule, and then they come in and cut it down. So remember, we start with the 45, and then we get the 18, the 5.8, and the 28. So its job is to help us process. And transfer RNAs, we know about these over and over again. So they help with us carrying our amino acid to our ribosome so that transcription can occur. Okay. And we'll stop there and talk about the other ones in the next video.